Hi, I'm Stephane Bertani from the laboratory of Dr. Frank Sauer in the Department of Biochemistry at the University of California, Riverside. Today we will show you a procedure of chromatin immunoprecipitation. So let's get started.
This procedure begins with formaldehyde cross-linking of human embryonic stem cells. Prior to cross-linking, human embryonic stem cells are thawed from liquid nitrogen for 5 minutes at 37 degrees Celsius and then washed with media. The cells are then plated on gelatin-coated 6-well plates and allowed to expand for 2 to 3 days. For each immunoprecipitation reaction, we will need to start with 50 to 100 million cells. The cells are first trypsinized and collected in suspension. Fresh formaldehyde is added to each cell suspension at a concentration of 1% and incubated for 10 minutes at room temperature. Next, add 1/10 volume of 1.25 molar glycine to quench the formaldehyde and incubate for 10 minutes. Rinse the cells twice with 10 milliliters of PBS and then pool them in 50 milliliter conical tubes. Spin the tubes in a centrifuge at 1000 RPM for 5 minutes at 4 degrees Celsius in order to collect the cells in a pellet. After the spin, discard the supernatant and resuspend the pellet in 1.5 milliliters of lysis buffer. Finally, transfer the cells into a 1.5 milliliter microfuge tube. After the cells have been transferred, flash freeze them